Hey, good afternoon everyone. So let's have another topic on cell cycle we call the cell division. Okay, cell division is another activity of the cell that helps the cell to replace the worn out cell. At the same time, this is another way to reproduce of another kind. Now, in cell division, it involves mitosis. Mitosis consists of 6 stages. Actually, this is a cell cycle already. So we have the interphase, prophase, metaphase, anaphase, and telophase. In the interphase stage, it consists also 3 major phases. We have the G1 and G2 phase and the S phase. So, the interphase will set all the activities of the cell, metabolic activities, and other functions of the cell. And while a small uh, time allotted only for the mitotic division of the cell. Now, G1 and G2 phases, these are known also as the growth phase, while the S phase is known as the synthesis phase, in which the DNA of the cell is preparing for mitotic division. So the DNA synthesis happens. Now, after the mitotic phase, this is the G1 phase called growth and normal metabolic roles of the cell. Then, followed by the DNA replication under the S phase. And we have the growth 2 in which the cell prepares for mitosis. So, this is the secondary growth phase. So, what happens during the first mitotic division? Okay, the prophase is the first stage of mitosis. The chromosome condenses and becomes visible. So, the diagram on the right corner, this is the chromosome of the cell. It becomes visible and it looks like in one haploid. So this is prophase, the first stage of mitosis. The chromosome condenses and becomes visible. The centrioles form and move toward the opposite ends of the cell, forming the poles. The nuclear membrane of the nucleus disintegrates or disappears, and the mitotic spindle forms, and the spindle fibers from each centriole attach to its sister chromatid at the kinetochore. So this holds the chromosome together in preparation for what stage? To prepare for the metaphase stage. Now, the metaphase stage, the centrioles complete their migration to the poles. The chromosomes lines up at the middle of the cell called the equator. So this is happen when during the metaphase stage. So you have there the poles. So these are the opposite poles, and the imaginary lines or the faded lines that you see are the spindle fibers derived from the centrioles. So the chromosomes are aligned together at the equator. The next stage, the anaphase. So the spindles attached to the kinetochore begin to shorten. So this exerts force on the sister chromatids that pulls them apart. So the spindle, as the spindle fibers attached to the kinetochore shortens, pulling the chromosomes towards the center, the centrioles or different poles, the chromatids began to migrate towards the pole, to the opposite pole. So spindle fibers continue to shorten, pulling the chromatids to opposite poles, and this ensures each daughter cell gets identical set of chromosomes. So what happens after anaphase? Now, when the chromosomes are already at the opposite poles, the, the, the next stage is the telophase stage. So chromosomes decondense, the nuclear envelope forms. So, hinahinay, the nuclear envelope start to develop. Cytokinesis. When we say cytokinesis, this is the division of the cytoplasm. So cytokinesis reaches completion, creating two daughter cells. So came from one cell, after mitotic division, it produces two cells. Now, we have another type of cell division, we call it meiosis. So we have different terms under meiosis. We have the allele. Allele means alternate forms of the same gene. Homozygous, having two identical alleles of the given gene. Heterozygous, having two different alleles of the given gene. Genotype, these are the characters that are not expressed within the organism. While the phenotype, these are the expressed traits of the organism. Example of a phenotype, if you have long hair, curly hair, brown complexion, a fair complexion, those are the phenotypic characters that are expressed at present. So meiosis is a special type of cell division that occurs in sexually reproducing animals or organisms. So the sex cells are called the ova and the sperm. So meiosis, the type of cell that undergoes are the sex cell or the gamete. So meiosis reduces the chromosomes by number by half, enabling sexual recombination occurs. So meiosis of diploid cells produces haploid daughter cells, which may function as gametes. So gametes undergo fertilization, restoring the diploid number of chromosome in the zygote stage. So when the egg meets the sperm, that's the time they become diploid again. So the ova always carries a haploid chromosome, while the sperm also carries haploid chromosome. So meiosis and fertilization introduce genetic variation in three ways. So this is the time that we became different from each other, even though we belong to the same species. 1. Crossing over between homologous chromosomes at prophase 1. So, during prophase 1 in meiosis 1, there's a crossing over of chromatids. So, that makes us different from each other. Second evidence for being unique from each other, independent assortment of homologous pairs at metaphase 1. So each homologous pair can orient in either two ways of the plane of cell division, and the total number of outcomes is 2n. So n is equal to the number of haploid chromosomes. Next, random chance of fertilization between any one female gamete with any other male gamete. So, what is the role of sexual reproduction in evolution? So, sexual reproduction in population should decline in frequent relative to asexual reproduction. So, asexual reproduction, no males are needed, all individuals can produce a new offspring. Sexual reproduction, only females can produce offspring, therefore fewer are produced. Sexual reproduction may exist because it provides genetic variability that reduces susceptibility of a population to pathogenic attack. So, the stages of meiosis can be broken down into two main stages. We have meiosis 1 and meiosis 2. Meiosis 1 can be broken down into four substages: prophase 1, metaphase 1, anaphase 1, and telophase 1. While meiosis 2 can be broken into four substages also, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Actually, in meiosis, we have two sets of mitotic division. So we have these important events. So prophase 1 is the most significant process of meiosis. You have to remember that one because this is the event where makes us different from each other. So variability occurs. But in prophase 1, it undergoes several stages. We have the first stage known as the leptothene. So this is the first phase of the prophase 1 of meiosis. Meiosis 1, so this is the phase thin threads become visible and homologous pair of chromosome approach each other. So this is the leptothene. Second stage of prophase 1, zygothene. So zygothene formation of synaptonemal complex, pairing a homologous chromosome. So there's a pairing of homologous chromosomes. So they become near with each other, start to pairing. Next stage, we have the pachythene, in which the crossing over occurs at this phase. So segments are exchanged. So there's a segment exchange in each chromosome. Then next stage, we have the diplothene. Okay, diplothene chromosomes get separated from each other. Synaptonemal complex breaks, but chromosomal pairs remain attached with each other at the site where the crossing over occurs, and this point called chiasmata. And diakinesis, the chromosomes completely get separated and the exchange segments become visible. So that is in summary what happens during the prophase 1. Then during the metaphase 1, microtubules grow from the centriole and attach to the centromere. So the tetrads are lined up along the equator. Then in anaphase 1, the centromere breaks and the homologous chromosomes separate. Noted that the sister chromatids are still attached with each other. So cytokinesis begins. So at the first stage of meiosis 1, we have the chromosomes are not yet haploid, so they are still diploid. So telophase, the chromosomes may decondense and the nuclear depends on the species. Then cytokinesis reaches completion, creating 2 haploid daughter cells. Okay, telophase 1, the homologous chromosomes pairs complete their migration to the opposite poles. So as a result, that is the result of the shortening of the spindle. So the haploid set of chromosomes is at each pole, with each chromosome still having 2 chromatids. So a nuclear envelope starts to appear surrounding the chromosomes. Then cytokinesis follows. Now let's have the prophase 2, which is the part 2 of meiotic division. So centrioles form and move toward the poles. The nuclear membrane dissolves. Metaphase 2, the microtubules grow from the centrioles and forming spindle fibers that attach to the centromere of the chromosomes, then aligning all the chromosomes at the equator. Next, we have the anaphase 2, in which the centromere breaks, the sister chromatids pulling them on each pole. Then as the chromatids pull towards the pole, the cytoplasm started to undergo cytokinesis. Then in telophase 2, the chromosomes may decondense, depends on the species. The cytokinesis reaches its completion, producing 2 new daughter cells having 4 haploid chromosomes. So if we're going to compare mitosis and meiosis, And here are the differences and the similarities. So, based on uh, organism, it occurs in animal, meiosis occurs in animals, plants, and fungi. And mitosis, it occurs all organisms except on bacteria. So, it occurs in all organisms except in virus and bacteria. It creates body cells. Meiosis creates gametes. So, mitosis produces somatic cells or body cells, while meiosis produces gametes. In terms of number of division, mitosis have one division, while in meiosis we have two sets of division. And in mitosis, there's no recombination happens. So, whatever the identity of the mother cell, it's also the same identity to the daughter cell. So, no variability happens in mitotic division. While in meiosis, there's a recombination or crossing over of chromatids that make us different from each other. And in mitosis, it produces two diploid daughter cells, while in meiosis, it produces four haploid daughter cells. Now, daughter cells are identical in mitosis in terms of quality, function, and quantity, while in meiosis, the daughter cells are genetically different from each other because during crossing over, each chromatids are different already from each other. And what are the similarities? First, it creates new cells. Follow same steps, begin with one parent cell. So we have also the same stages, like it started from the prophase, metaphase, anaphase, and telophase. So that is cell division. Thank you for listening. Bye.